Today is the 9th lecture of our online school on applied population genetics. And our lecturer today is Thorfinn Korneliussen from University of Copenhagen. So Thorfinn, welcome.
Yes, thank you. Last time I talked about the ancient DNA, and then there was a brief introduction to sequencing data that Russell Corbett already talked about. I extended this with how to define and how to calculate a genotype likelihoods. So there was a question that I got over email that I think I'll answer first. So let me share this. So Vladimir, can you give me a share control here?
Yes, of course. Just a second.
Good. Okay, let's see this one here. Good. So when we are sequencing ancient DNA, then a difference from modern is that it's short, that the fragments are much shorter. We have very little biological material to work with, so we are not able to obtain so many reads that would be possible if it was modern. And then we have this telltale sign of deamination, a damage signal, which at the 5' end means that C turns into a uracil, and when we sequence this, it's being interpreted as a T. That is from one direction. If we were looking at the 3' end, then we would get a damage signal, which would be a G to A, and that is to some extent what we are seeing here. What we are, one thing also to note is that we have this excess. It looks like there's an excess of G to A here in the very beginning, and this is something we would expect if we're looking at the overall proportion of A, C, Gs, and Cs along the read. Then we always see that there's some jittering in the beginning, and this is most likely due to how the chemistry work, how the libraries are being built, these enzymes, how they're operating. Another thing is that there are many different protocols in the lab. In some instances, if you have your double-stranded DNA, then there might be a single piece of your backbone that gets lost. We call this a nick. And then when we try to sequence it, then we have to repair this. And as soon as it hits a nick, then it discards the rest of that backbone and is then using the what's on the opposite strand, doing the reverse complement for extending the other backbone. So it's a very elaborate process, and sometimes we cut off the damage and from the overhangs when we see it, and the other times we repair it. So it's not a straightforward clean answer. What we do want to see is that we have an excess of C to T and G to A. Of course, if it's a very, very low coverage data we are working with, then it might not actually be possible to see this kind of damage signal. We require many reads for being able to see this. In practice, how we so I just said that it's possible to see to observe that a C turns into a T. How we do this in practice is that we take our read, then we align it against a human reference, and then we look at what the reference is at that position. So if it aligns to position 700 on chromosome 1, then we look at chromosome 1 position 700. And then we're seeing what is the reference. Is the reference an A? Do we observe an A in our sequence? Okay, then we write down that number. We do this for each position in our read. Actually, we do this from both ends, from the 5' and to the 3'. So this would be a sort of a 4x4 mismatch matrix for each position. And this is how we are actually seeing that, or saying that we have damage. Of course, it's not really a damage, right? It's excess mismatches that we interpret as damage. If there had been no sequencing errors at all, and there had been no natural variation, then we would only observe the numbers in the diagonal of this 4x4 table. Okay, so this is what characterizes ancient DNA, and when we are sequencing it, then we get very little DNA out of it, very few reads. So might have a high clonality, and that limits how much useful data we can get out of it. We measure the amount of material that we are interested in, and we call this the endogenous content. And this would be a percentage that we can use as a scaling factor and compare to what we would need if it had been a modern sample. So if it has 1% endogenous, then we would need to sequence 100 times as much if we had been sequencing a modern genome. Okay. Okay, so that was the first part I talked about last time. So I'll go back to the NGS part that we didn't finish last time. This one here. So I've put all the slides that we might or might not go through today on the chat. So feel free to look at them and go through in your own tempi. That's fine for me. There's a lot of nasty math in the EM algorithm. Don't worry, we won't go through this. We'll only talk about this at the intuitive level. So here, can everybody see this?
Yes, sure.
Oh, good, fantastic. I'll try to make this full screen. Okay, so again, so I think both Rasmus and Ross and me phrased these things and defined these things, right? So we have an allele. An allele is a variant form of a gene. We have a genotype. Those are the alleles for specific individuals, for a specific individual. And then a genotype calling is to figure out what the actual genotype is for a specific individual. And this is what we cannot do with sequencing data unless we have very, very high coverage. Another thing I didn't, that might also should have been explained here, is the process of a SNP calling. So SNP calling is figuring out, say you have a sample from a population of 50 individuals, then it's figuring out all the positions that are variable within this subset, right? So you want to figure out which side are variable. We call this SNP discovery or SNP calling. So SNP calling is figuring out what the variant sites are. Genotype calling is figuring out what the actual genotype is for a specific individual. And a genotype consists of alleles. So this is, most of what we are doing end up being focused on at the base level, right? So we have a nucleotide that can be either A, C, G, or T. So we call this an allele because these are SNP variations. And since we are diploid one chromosome from our father and from our mother, then the allele consists of two bases. This is what we've been doing a lot in humans with regards to sequencing data, but there's a whole family of variations which are called these structural variants like insertions and deletions that we won't talk about for this. To my knowledge, that has really been a useful probabilistic model for insertions and deletions developed yet. Okay, so this is what we have. Now we defined it again, and hopefully it's clear. We cannot do a genotype. We do not know what the genotype is. And instead of trying just to hand wave and see what might have generated all these T's and C's, then we are calculating a probability for the 10 possible genotypes. Okay, so let's see here. So if we have 4 bases, then we have 10 different genotypes because the ordering does not matter. The ordering does not matter. We cannot distinguish between A, C, and CA. So when we are looking at a genotype in this context, we do not know if the A belongs to your mother or to your father. Okay, so these are what we call unphased genotypes. We can also start to look at phased genotypes. Then we know which allele originate from our mother and father. But this is difficult to do with the sequencing data. Then you need to estimate this in a probabilistic context. This is called there's a whole family of these called phasing algorithms. So we are calculating these genotype likelihoods. So this is the probability of our data given a genotype. And here we are using this GATK model, where we are assuming that there was no errors in the quality score. We can trust that completely. And then that there are many different genotype likelihoods models. And then there was a full example of how to do this. So hopefully it was clear how these genotype likelihood was calculated and what they were supposed to represent. We are assuming each reach is independent. This is why we can just take the product here to obtain the genotype likelihoods. Good. So for the example we had all these bases. I think it was 2 C's and 9 T's with these ASCII encoded quality scores. These gets transformed into a proper quality score by taking the ASCII value and subtracting by 33. We convert these quality scores into error probabilities. So if we look at this one here, the T that has a quality score of 22, this means that it has a probability of being incorrect of 0.6%. So the probability that it is correct is of course 1 minus. So the probability that it's a C. Now there's this, we are observing a T. So there are 3 other options that it could be either, and we assume that these are equally likely. This is of course an assumption which is clearly wrong, but in practice it seems to work quite well. These are one of the things that we are trying to model better. So we are not assuming an equal probability that gets distributed to the unobserved alleles. So we have all these. These are for the different reads. Okay. So all what we have here is not for the same read. It's from different reads. And it's for different reads that overlap a specific position. So it would be this example here where we have the first read that starts very much further before the rest. Then then we have 5 base pairs where we don't map any data to. Then suddenly we have a new read that maps to this position. So overall, when we are looking at this side over here, then all these bases belong to different reads, and they have different positions within that genome. Okay, so if there was, like in the context of ancient DNA, then you know that there's excess error in the beginning of the read of C to T. Then the C to T damage would be much more likely to be in the beginning of each read. So when we are modeling the genotype likelihood, if we wanted to do this properly for this situation, then we didn't really care about modeling the ancient damage in these reads where we are in the middle of the read, but it might be useful to actually have this T. That might actually have been a C that was converted into a T. Okay. So continuing this example, all these are different reads. So we have these probabilities, and then we can just plug them into this GATK model. Here we are assuming that we are giving them equal weights. So, but these are just scales to a constant, so we could discard those if we wanted to. Of course, this is, if you have very high coverage data, then these probabilities turn very small. So it's useful to switch things into a log scaling. And this is what we see from our example on the left-hand side. These are the genotype likelihoods for the example. Intuitively, we can see that the ones that are the highest, right, the ones that are most likely are the CT and the TT. The CC is not that likely from this example, but we can see that it's this one that's these two that are the most likely. And if we shift things into a normal scale by just taking the exponential or whichever log scaling we were using, then we get these values in normal scale. And there we can see that the TT genotype is 10 times smaller, less than 10 times smaller than the C2T genotype. Okay, so the whole problem of what we want to do is to try to figure out what genotype each individual has. If we have that information, then we can do everything we wanted. Then we can use all the fancy algorithm that population geneticist has been developed for the last 40 years. And we can even assume sometimes that if we have a phased genotypes, then we can do even more elaborate downstream analysis. But getting the genotype is something we cannot do. And when we try to call genotypes, then we are likely to bias our results, and we need to apply certain kind of filters. So the read distribution for a genome, we had an example here. We have very few sites with 2x, and we have quite a lot with 6x, and then we have a lot with higher coverage. So it's not, the depth is not constant along the genome. So whether or not we can call genotypes, but should also depend on the actual number of reads that we are basing this hard call on. So let's go through an example of how to call genotypes.
Excuse me.
So, yes?
May you switch to the previous slide?
The one we saw before? With
yeah,
the
this one. No. This one. Oh, no, not this one. With likelihoods and log likelihoods.
Okay, so it was this one.
Yes. Why on the left, all the numbers corresponding to zeros are different?
So it's when we take the exponential of this, then it turns into a very, very small value, like 10 to the power of minus 12. And then for a slide, we're just saying that it's 0. Of course, if we had, it was because it would obscure the slide.
All right, it's just rounding, right?
Yes, it's just rounding.
Thank you.
Yeah. So actually, what we also do in practice is that we are shifting things to a log, but we are not. We're actually using log likelihood ratios to the most likely. So we take all these genotype likelihoods, and then we subtract by the maximum. If we do this, then this value will be turned into to 0, and then the rest of these values will just be shifted towards 0 with a value of 8.31. Okay, so this is what we do in practice, because we are not really interested in the actual value of the likelihood. We are interested in the ratios between the likelihoods. Okay, so we want to do genotype calling. And then this is some of the different genotype likelihoods we talked about. So of course, the most simple family of how to call genotypes is just to pick the one that looks the best, right? The maximum likelihood. And in this case, it would be the C to T. We can do this, and it might work and it might not work well. It depends again on the context. A much better way to do is to ensure that the difference between the two best ones, the two most likely ones, are large enough. Okay, this is a much better approach to use. Of course, it's what's even better is try to calculate a kind of posterior probability, so we can interpret this. And then if we have a posterior probability of the different possible genotypes, then we can say we only call a genotype if we are more than 95% certain that it is correct. And then you would use this in combination with we want to have at least 4x sequencing depth, and then in practice, you apply many different kind of filters to avoid biases from the sequencing platform. So if we wanted to calculate a posterior probability of the 10 different genotype likelihoods, then we could just assume that they were equally likely. Okay. So we just multiply these with 1/10, 0.1, and then we normalize, and then we get this posterior probability. Okay, so this is the most simple way, and this is we have 10 different genotypes, so it might not be a bad approach to do, but we can improve this dramatically. Well, what we can do is that we can start to look at what the reference is. So if we know that the reference that we are aligning against is highly curated, so this is very likely to be correct. Of course, it's a haploid representation of a diploid organism. So there might be so it's of course not the best approach, but it should give you a lot of information just by looking at what the reference is. So what they did in some of the first sequencing projects was to look at what the reference is, and then they were given that a weight. So if the genotype had an allele that was corresponding to what the reference was, then they would give it a probability of 0.99. And I think this was used in the first Asian genome that was sequenced. And again, so when you do this, then if you do this in this case, then you dramatically change which call genotype you're using. If we use this 1/10 prior information, then the called genotype is a CT with a 92% chance of being correct. On the other hand, if you're using a different kind of prior where we put a higher weight on the reference, then it changes, and then our call turns into a TT with an 80% chance of being correct. So the prior is changing a lot. Another approach, which is what is used in practice, which is, let's see, yeah. So this is what is used in practice. And this is, instead of trying to look at the single individual, instead of just looking at one individual and then try to figure out what is the genotype that we observe in this individual, then you can actually look at the information for all individuals. Say you have 100 information that they're all with low coverage, then you might have a single individual where you observe 1 C and 2 T's and 3 G's and 0 A's. And then in the next individual, due to sequencing error, you would also get some randomness, but what you would overall have would be a very good representation of what the two most likely alleles would be, right? So you can start to elevate the information from multiple individuals to multi-sample joint calling. And the most common thing to do is to calculate the frequency of one of your allele across all your individuals, and then use that as a sort of prior to your in your analysis under some assumptions like Heidi Weinberg and these things, right? So this is what they do in practice, and if we apply this kind of prior for this example, then we'll end up with two very likely genotype calls, right? The best one has a probability of 56%. So so if you were running this analysis, what would your conclusion be from if you wanted to call genotype for this based on the information you had here? Would you believe it's possible, and which one would you trust the most? So it's of course not, if it had been, if all different genotype calling approaches had shown the same, we should be able to trust it. But here that we're given so different results, we would completely discard this genotype call, because none of them is really reasonable. And this is also sometimes what we do in practice if we are getting some strange results in a downstream analysis based from an NGS project. Then if it was based on called genotypes, well, then we'll try to fiddle a bit with the genotype calling approach, and we might even try a completely different software to see how different these genotype calling approaches are. So in a different kind of exercise where we had where it had been more like class teaching, people sitting in front of the computer, and we had walked around, then you would have been asked to do genotype calling using 3 different approaches, and then do Venn diagrams to see how different they are. Because of course now we're just talking about genotype calling. There's also how reliable can we call genotypes if we're using the frequency as a prior for the very low frequency variants. This is one of the things that people have invested a lot of time in, right? Rare variants. This would be the singleton category of your site frequency spectrum. These are the ones that are the most difficult to call. Okay, so in practice, if you're working with human data, then you can then there are fundamentally 3 different approaches that you can apply for doing this kind of genotype calling. Here in what we call the single sample, that would be like the maximum likelihood estimate, where you're only using information for one individual. Then the second approach is where you are starting to pull in information for multiple individuals, right? You're calculating a sort of prior across all individuals. You're determining what the 2 segregating alleles are. So segregating means the variance that we observe across this position. So it would be a matter of just figuring out what the 2 most likely alleles are. This is not easy if you're working with rare variants. So if you have 100 individuals and you wanted to calculate the site frequency spectrum, then the singleton category, that is the single heterozygous call in a single individual out of all 100 individuals. So it would be 1 allele out of 200. So this is very difficult to call with many approaches, because you don't have much information in your prior. On the other hand, if it's a variant that is segregating at 50% in your population, then it's very easy to both do SNP discovery, and then the prior also has more information. Because of course it's how to calculate the frequency. That's the topic for in 2 slides, but that's by itself is also tricky. But the final approach that is much better if you have the possibility of doing that is to start to use like reference panels. So if you have a representation of the haplotypes that you observe in a population, then when you look at your sequencing data, then you can try to figure out, okay, with this combination of alleles, then I'm very likely to have these, I come from this reference haplotype. So if you do this, this is of course cheating to some degree because now you're using external information. The other approaches were just using internal information, well, apart from the reference that was used as prior. But now you're starting to use external information, and by doing this, you also get a lot power. So this is a classic, almost like a rock curve. A rock curve. So what we're observing here is the proportion of non-calls, and then we have the genotype call accuracy. So the best approaches would be the one that are on the top all the way. Okay, if you want to call everything, right, that would be the one that might even have a very low probability, posterior probability. Then these would also be the ones that were wrong. Of course, how to generate a plot like this is not trivial because you need to have a true data set, right? You need to have a data set where you actually know what the underlying genotype is. And in the past, there's been like repositories of highly curated Sanger sequenced genotypes that has been used for figuring out what the truth is. And this data set is actually very representative. It's from a huge consortia called 1000 Genomes from their pilot project from a 3 to 4x the depth sequencing. And this is only the common variants. Okay, so of course the question is, why don't we just always call genotypes using this kind of haplotype information from reference data? So well, it's not always possible. You might be sequencing a species that's never seen before, right? It might be a special lizards from somewhere in Africa or Australia. Then you don't have this. Then you need to resort to these more primitive approaches where we are not having this information. Okay, so that was genotype calling. Then so let's change topic a bit. Now we know what genotypes are, we know what alleles are, and we know how to call genotypes. Let's also talk a bit about frequencies. And allele frequency, that is in this context here, it's the relative frequency of an allele for across all different alleles. So if you have, we are assuming that only two alleles exist, then it's just a proportion of one allele divided by all the alleles. Okay, this is how we define the allele frequency. This is what we see here. Of course, in practice, there's this thing that we need to figure out what the two alleles are. But that's a different topic. But assuming that we know what the two bases are for each individual, that we have the hard call of the genotype, then we can simply calculate this frequency. Okay, so here is there is an example. This is very, very similar to what we had, what I asked you to do the last time. This one here, to calculate the frequencies, how many times do we observe an A across all the different ones? Okay, so let's go back here. Okay, so to begin with, when we were working with sequencing data, we didn't really have a strong mathematical foundation for calculating these things. Genotype likelihood wasn't even invented or being used widely. So what we ended up doing was to for each site, for each individual, then we would calculate the we would look at our sequencing reads, and then we would say how many times do we observe A, C, G, and T, and we would call those like the count files. And then based on those, then you can come up with these elaborate frequency estimates. And these are, of course, in the example before, there we are using the, then we are just using the hard calls. In this example we have here, then we are looking at the sequencing data. How many times do we observe an allele 1 in one individual? So this would be the number of bases, the number of allele 1 bases for this individual. And then we would well, then we would get the frequency, and this example we have here is the most basic one, where if you write down the equation and conditional on that an allele has changed with a certain probability, because the sequencing data has probability, then you can derive this estimator. This one here is based on the same idea, but what we are taking into account here is that there might be a large difference in sequencing depth between individuals. So we should take into account that individual 1 might have many reads, but the next individual might not have that many. So this is what this estimator was solved. But we also have this thing like if we're assuming that we have genotype likelihoods, then we can use the genotype likelihoods to calculate the frequency, and then suddenly things change in from a statistical model that is based on the hard counts from count data. You have that many A's and C's and G's, and suddenly you're modeling things in a probabilistic framework. So we are going to talk a bit about that. I'm going to show how these things are applied with regards to the EM algorithm. But this is, of course, a much better approach. And the reason why it's better is that when we were using the count of the data, then we were discarding the quality score information. So we lost all information about how much do we trust that it was an A that was sequenced. If we are just using the counts, then we are saying, okay, we have an A, so we are losing that information. If we are using that to have genotype likelihoods, then we have a much better way of knowing how wrong we are, right? Doesn't matter how wrong we are measuring things, as long as we know how wrong they are. So here we are plotting two things. On the y-axis, we have the true genotype frequencies. So those would be the frequencies that is based on the known genotypes that we trust with high credibility. And then on the y-axis, we have the one that is based on the count data. Okay, so if we had a perfect match, everything would be straight on the diagonal. But we can see here that they are actually, it looks pretty good, right? That it's not that far off the diagonal. But if we change into this kind of maximum likelihood approach, where we are using the full information of the data, assuming that genotype likelihoods contain the full information of the data, then we have improved this drastically. Okay. So assuming that we have a way of, okay, so this was how we are calculating frequencies, okay? Genotype calling, frequencies, now SNP discovery. How do we figure out if we have a variable size? If we had the perfect genotypes without errors, then we can just say for every position where we are observing more than one allele, then it's a variable site. Then we're saying that it's a SNP site, right? It's a variable position. this is not this we cannot really do with if we have sequencing data, because we might have low coverage. So what we can do is to use the frequency as a kind of information to base the SNP genotype calling, the SNP calling on. And this would is in practice it's a very good approach. If you have 50 individuals, then you have 50 diploid individuals, then you have 100 alleles. Then you would say that all the variable sites are those where we have an allele frequency above 1%. This we could do in practice, since this is the allele frequency estimation is an optimization that, and in practice we're using an expectation maximization algorithm. Then things turn very they never really gets all the way down to 0, but very close to 0. Okay, so what we can do is to define a kind of likelihood ratio test where we have two models, one where we are assuming that there is no variability, and one where we have assuming that we have an allele segregating in the population with some frequency. And then we can look at the likelihood, the difference in these likelihood, and this is a likelihood ratio test, which is chi-squared distributed. And then we can do this corresponds to a p-value, and then we can say that, well, if we are interested in general population, demography, things that are informative across the entire genome, then we can just use as many sites as we almost want. But if we wanted to have very stringent criteria, like a very small p-values, then we would only allow those that had very high frequencies. Okay, so depending on what you want, you might want to include different thresholds for when do you call a SNP in this kind of probabilistic approach. And of course, so this is the EM algorithm. We'll talk a lot about that afterwards. So I think I'll skip this. I don't think this might be that motivating here. Yes, what I would like to do now is to go through some very few exercises. I think it's like 4 lines that I'll just go through. You can see the commands that I'm running here. And this will just show you how to calculate allele frequencies from data. Okay, so I'll kill this slideshow now, and then I'll open a terminal. Are there any questions for this?
You will not need any breakout rooms for that, right?
No, so this would require that people download some software.
Yeah, sure, we are not prepared for this, I guess. Not for now. Okay.
No. Yeah, so what people can do is that I'll go through this, and then if people are interested, then they can run it for these themselves, and we can talk about them again at the beginning of the next lecture in a week. Okay, so I'll start so I haven't really prioritized exercises, given that it's going to be a bit difficult to actually supervise how people are doing, even if they are in these breakout rooms. So let me find this here. Okay, so so first off, just to get things started, then the purpose here is to calculate frequencies using SAM tools so did Ross talk about, did he give you a tutorial on how to use SAM tools and BCF tools and these things?
I'm not sure that everyone who is present here was present.
Okay. So
No, I think Russ
don't
didn't
actually
show
know.
that. Russ didn't show some tools, I think.
Okay.
Not all of them, I guess.
Okay, so this is actually very important because, okay, so if you're working with sequencing data, then there are some tools that you cannot avoid to use, and SAM tools is one of them. So it's called this sequencing alignment format tool. It's written by a person that originally came from China called Heng Li. He was in a big genome center in China called BGI, and then he was a postdoc at Richard Durbin's lab, and then he moved to the US and was part of David Reich's lab, and I'm not sure where he is now, but he has made tremendous amounts of very, very useful tools for analyzing sequencing data that are used everywhere. And one of the first one was SAM tools, and SAM tools is a tool for manipulating and doing basic analysis of SAM, BAM, or CRAM files, right? These are the same kind of files. So this, in the beginning, there was SAM tools, and then they could see that it was a better way to organize their software was to separate it into 3 different tools. One is like a general library called the HTS-lib, which allows you to use their file passing API, so you don't have to write your own parser if you want to access sequencing data. This is very, very useful, and that has really changed how easy it is to write software that is using sequencing data. Then you have a software called SAM tools, and in the beginning, SAM tools did all the analysis itself. That was the only software, but now what SAM tools is that it's a software for converting and manipulating single SAM or BAM or CRAM files. If you want to do the actual analysis, like population genetic analysis, whole genotypes and SNP calling, then you need to use BCF tools. So if you ever are working on this kind of sequencing data, then there's no way to avoid these tools. Okay, so first off, I'll just copy in code as comment so people can see what I've done here. So just a second. I'll share my terminal. Hopefully it's not too small. Let's see. Can people see this?
Yes, sure.
Yes.
Good. Okay. So first off, what I do is that I use all these commands for pulling the software and data from GitHub. And GitHub is also one of these online data repository places where most of the data is nowadays. And it's very useful if you're a software developer because it helps you keep track of all the edits that you have made. Then I'm downloading all these software. Okay, so to begin with, I have a small software that I did during my PhD called Angst Analysis of Next Generation Sequencing Data. Then there's a folder repository that contains BAM files. Then we have this HTS-lib, SAM tools, and BCF tools. And then I'm compiling all these using these kind of arguments, right? I'm stepping into the HTS-lib, I'm writing make, and then I step back to the current location. Hopefully this is clear for everybody. So if I go into this small BAM folder and look at all the BAMs, then we have a list of the BAMs. These are compressed, so you cannot view them if you do less and then copy in this. It will be it just looks like random crap. It's because it's compressed and it's binary, so it's structured data. If we wanted to actually look at this, then we could use the SAM tools command. We can do view. Per default, if you do SAM tools view, then it will take whatever file that you are supplying it with and will show it as a SAM file. So I'm piping this into less with -sn. S mean that it makes it it doesn't clip. Well, it doesn't fold your text. It just clips it, and then n just gives us a line number in case we need to talk about specific lines. So if you do this, this is how it looks. And this is, I'll just extend this a bit to the left so we can see more. The line number, which is put on by the less command, the read name, then we have the flag, and we have the chromosome and the position. We have the mapping quality, we have the cigar string. We can also see here that it's a paired end read, and this, it means that it maps to the other read, right? You're sequencing both of them. It says that the other one is mapping to the same chromosome and has this position. It gives us this value here, which is like the insert. The insert, this is the original biological molecule where we are putting adapters on both ends. So this is our fragment length. If this had been ancient data and we did paired end, then all these would be very, very small. And you can see most of these are around 300 to 500, which is which is common to have it in that range. Then we have the actual sequence basis. This would be the raw, uninterpreted output from the FASTQ file. And then we have this, the actual quality score. So these are the quality score that contains the information of how much we trust the basis. We need to look at the ASCII value, subtract by 33, and then use this this log 10 style scaling to get the probability. Then we have all these extra tags in the end. These are something that the aligner and downstream tools might add to each read. So in this example, we have the read group. This is to keep track if your SAM file is emerged from different sequencing runs, then you can keep track of this with the read groups. You have the NM tag. This will tell you the I think it's the edit distance, like the number of changes you have in your sequence to your reference. Then you, yeah, there are many of these. Most relevant, you have something which is called the BQ. This is, in the beginning, it was, when you were working with sequencing data, it was found that that there was a lot of mapping problems around insertions and deletions. This is what caused problems for the mapping. So what they did was to refine the quality score such that it's, if a read, if you have a base that's very close to an insertion or deletion, then you can downgrade your quality score so you trust it less. This is called back recalibration. So what you have here is the difference to the original quality score. This is something you would nowadays not keep in your data set. You would actually compute these on the fly because it's so fast, and it's also a lot of redundant information. And one of the biggest problem with high throughput sequencing data is the storage capacity. In our sequencing center, we are looking at over the next 3 years that we need to have 2 petabytes of storage just for storing the raw data, right? And then there's all the extra storage you need for actually analyzing it afterwards and making copies, converting fast queues to BAMs and CRAMs and generate genotype likelihoods. So storage is a big thing, a big issue. This is also the reason why they have changed into the CRAM format. So the SAM is a text format. Then there's a binary representation called BAM. It's good on 2 accounts. First off, it takes up a lot less storage. It has a less storage footprint, but then you can also index it. So if you had a file with this, one thing to notice is that this is a sorted SAM file. So you can see that each read starts at the same or position that's higher. So it's been sorted, and there's this relative ordering induced by the chromosome and the position. So you know that the position of chromosome 2 would be after chromosome 1. Position 2000 would be after position 1000. So you can create a sort of index. So if you wanted to go directly to chromosome 17, position 2 million, that you have a favorite gene that you want to look at, then you don't have to do a linear search throughout your entire text file. You can supply a command to the SAM tools command, and then you can jump directly to those regions to those reads that overlap a specific region. So this is how the SAM file is organized. Very importantly, what you don't get in the output when you are just viewing it is that you're not getting the header. There's this hidden header, which is very important also. The header part starts with an add, it has a version, and then it also tells you if the BAM SAM file is coordinate sorted. So here you have your SQ tag, that means your reference genome. That's chromosome 1, 2, 3, all the way down to the mitochondrial. And then we have some alternative chromosomes. We have many of those in this example. So the SQ tag defines reference names. You have the PQ tags, and this is, the PQ tells us about which programs that has actually generated the data that you observe. And the idea is that you can, if you get a BAM file, then you can actually go back and see, okay, who has manipulated this data? What analysis have they done? How have they transformed this data to, what have they transformed it from? And here you can see what they've done. They have run a BVA index and align, and they've done something called SAM to fixed BAM, which I assume is their internal tool for analyzing it. They're also using a GATK, and then they're doing a lot of fancy things, like they're doing a realignment around indel, similar to what the back does, and then they're also marking the duplicates with Picard, and then you can see which exact the arguments they've used. So this is a sort of log file that is put into the BAM file. Okay, so so this is how a raw SAM file looks when we take it from a BAM. If you switch into CRAM, then you're only storing the difference to the reference. Most of the genome is redundant. If you're sequencing 1,000 read, then most of those reads will actually be the same as the reference. So if you're using that intuition, then you can say, well, let's not store the actual read, but let's only store the difference to the reference together with the quality score, of course. This is the idea behind CRAM, and it's very useful. Of course, it's a bit more heavy on the when you want to compress and uncompress it, and you need to have your reference that you have been used for mapping it. You need to keep that. If you don't have that anymore, then your CRAM file could be corrupt, unless you embed it in it, of course. Okay, so in these, this was a brief introduction to SAM and BAM using SAM tools. So what we want to do is to we want to try to do basic SNP calling and genotype calling using BCF tools. And here I'm using BCF tools because that is the most common thing that people are using. And this is the command we are using: BCF tools, mPileup. mPileup means one of the first format was something called pileup, where you would essentially just you would slice up all your reads. So each line would there you would have the information that would correspond to one position. Okay, so this would be the pileup of reads for each position, together with your quality score and position and what the reference is, and you could also get information about which position within the reads you had. So if we apply this approach here, then we are saying Samswell, blah, 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 blah. We're also telling it what the reference is, because that will make it a lot easier for BCF tools to actually do the calling. If we run this, again, we pipe it into less with SN, then we get something which is not a SAM or BAM file anymore, but it's a BCF file. I'm not sure if Ross talked about that, so I'll also give a 2-minute primer into those, because that is the container format that people are using when they are storing variant information. In the BAM file, the header part consisted of the add. In the BCF file, then it starts with, the header starts with 2 of these hash marks. Then you have multiple definitions. Most relevantly, we have the commands that we used for what we have done now, similar to the PG tag in the with the SAM tools. And then with what our reference is, and then we have these, we store information about how long the different chromosomes are. Then the software will generate a lot of summary statistics that can be used for many different things. And here they actually define those. So they define something that it's an indel. There's a description, indicate that the variant is an indel. So when you get down to the variant part of the data down here, if it says indel, then you can go back in the header part and use that as a documentation for what it actually is. Then you have some filtering options. You have like a read position bias. So this will try to figure out if there's some kind of bias. If all the reads, all the bases that you have for a specific position is in the beginning of the read and it's not distributed along all reads, there might be a problem with the mapping. So this is the header part. Then you have the single hash mark. This indicates indicates that now the data part starts. We have these columns which has to be there: the chromosome, the position, ID. That's an identifier for the position. So if you at some point want to merge your data with some SNP-chip data, if you SNP-chip data there, each marker was defined by an identifier, not by a chromosome name and position, then you could put in the name. Then you have what the reference is. Here we have that it's an A. This here means that we don't observe anything else. We only observe A's. We have a sequencing depth of 1. And then we have all these things that have been calculated that I don't know what all of these means, but you can go up and then see if it's useful for what you're doing. Then you have something which is called the PL tag. That is the Phred scaled genotype likelihood. Okay. So if you look here, you can see that all the likelihoods are 0, 0, 0. This indicates that there was no information that they're all equally likely. So when we go through here, we can see that we have one over here. So there was one individual where we observed a different base. Okay. But the reference is the most likely, and then the two alternatives gets gradually less and less likely. Okay, the lower the value, the more likely. So this is the BCF file. BCF file are text files, they have the same problem similar to SAM files, that you cannot go to a specific position. So there's a version of VCF similar to SAM and BAM. Then you have VCF and BCF. So BCF is like the binary equivalent of these VCF files. So if we wanted to call things, then we would take the input that you just saw where we had the Phred scale likelihoods. Then we would take that output and again pipe back into BCF tools. We feed it back in, but using a different subroutine, which is called the call. The first one we used was called the mpilup. We do this. And then we skip the header part. And then we get to the actual CLS. Okay. Then now it gets a bit more interesting, because what we saw before was a lot of sites where there was really no information. Now we have done snip calling using the built-in criteria in BCF tools for doing snip calling. How that is defined is, of course, something to consider. So what we have now is that we have a reference and an alternative. Then we have a quality. And then we have a sequencing depth. And then we have all these values that might not be relevant, but you can see that there's a lot of filters that's being applied. Inbreeding coefficient test. This could be the sort of Heidi-Weinberg test. And then you have your, we used the call command. Previously, we only had the Phred scale, the genotype likelihood. Now we have a different information, which is called the GT. This is the call genotype. So for each individual, this is encoded like this. We have two kinds of information. It's separated by colon. First, it's the GT information, and then the Phred scale likelihood. So if the genotype is called 0/0, then it means that the allele is a reference/reference, right? So it's a reference homozygotes. It's homozygotes for the reference. If it had been 0/1, it would have been heterozygous. One allele is the reference, the other one is the alternative. Then you can go back to the column and then see what the allele was. And if it's a 1/1, then it of course means that it's homozygous for the reference. So here we are using slash in the VCF and BCF file. This can also store phase genotypes. Then it would not be a slash, but a pipe, a vertical line. Okay, so here you would actually get the genotype likelihoods and the genotypes, the called genotypes. So what we want to do is try to compare the allele frequency. How do we do this? How do we calculate allele frequencies? Well, we extend this command, but in order to speed up things, I'm using this parameter called /R1, and that means use the region or the chromosome called 1. So now we limit the analysis to just a single chromosome. This is a very small test data set, so it would of course be possible to do, but it wouldn't be worthwhile waiting for. And now instead of just looking at the output as a VCF, now I'm looking at the output, I'm generating a BCF file. That is the dash capital OB dash small o, and then the file name for the output. So now I have a BCF file in this directory where I'm working. Okay, so then I want to calculate allele frequency based on the called genotypes. Then again, we are using a BCF tools, and then they have a whole array of useful plugin commands that is not really core to the to BCF tools, but it's stuff that is based on the BCF tools functionality. So this one called fill tags will just fill in the most relevant tags. And what we are going to look at is the one that is called is the AF tag, which is the allele frequency based on the called genotype. So if we were to look at this, then we could write BCF tools, view. Then the BCF gets interpreted into a VCF file that is nice and viewable. Then we are looking for this thing called the AF tag. That's the allele frequency. It even has a math tag, the minor allele frequency. The minor allele frequency is just the smaller of the frequencies. Okay, so if it's diallelic, then the sum of the two allele frequencies should sum to 1, and then it's the smaller one. This is a kind of polarization we sometimes use also with the side frequency spectrum that we might not get to talk about today, but there you need to, everything has to be in relation to something, right? So when we're calculating allele frequencies, either you can say it should be for major and the minor, right? The most frequently and the second most frequent allele. What they do in SAM tools and BCF tools is to look at what the reference and the alternative is. Another thing to do is that you have an outgroup information. If you go back to what the ancestral state is for the different alleles, then you can polarize things according to the ancestral and derived. This is what you traditionally would do with the side frequency spectrum. But for these basic analysis, it doesn't matter. So we are interested in extracting the AF tag. So when we look at, let's say we look at a specific position here, then we can see very long within this, we have the allele frequency and we have the minor allele frequency. This would be horrible to extract, right? It would be a Perl script that passes this. It would be very cumbersome to do. So they have made this sub-functionality, which is very, very useful. It's called BCF tools query. And there you define which information you want to extract from the BCF file. So here we want to extract the chromosome position, the reference, the alternative, and the allele frequency. If we do this, now we're just using head. Then we extract those directly and get it into a format which is much nicer. We can actually define the format ourselves. Okay, so I have 1% power, so let's do a 5 minutes break while I get the my adapter, and then we continue. Okay.
Okay.
Okay. Should I start?
Yes.
Okay, super. Okay, sorry for this. I should have prepared a bit better, but now I have a turbocharger on my laptop. Okay, so what we get here in the output is that we have the chromosome position, then we have the reference and the alternative, and then we have this very strange reference. So this would indicate that we have a sort of insertion or deletion in our data set for this specific position. And then we have the reference or the allele frequency. So this is how you would do things if you were to use the BCF tools, which is what people are using most of the time. It's a very, very good tool. So here, now I'm not outputting the allele frequency, but the minor allele frequency. Then, so in this software that I've written together with other people, we made this software, started doing this many years ago. It was before the advent of HTSLib and all these tools, and we found that there was extremely little documentation about how things were implemented. And if you wanted to figure out how things were done, then you would have to look through the source code, and the papers wasn't published that described these things. So we ended up making our own file reader and then started adding functionality into this because we were interested in developing the tools that we were going to apply to for this low coverage data. so that was how it started. So it works a bit differently. We define the list of files that we want to use. We define which kind of major and minor we want to use. So the idea here is that if you're running the other tools, then it's there's really no way of defining alternative models, right? There's one genotype likelihood model. There's one way of specifying what should be major and minor. Actually, BCF tools has 2 versions now. So what you can do in Angst is that we wanted to compare what was best. And then in order to do a proper comparison, you might as well just implement everything into the same framework. So if you wanted to see which kind of genotype likelihood models you would have, then you would run it like this. Angst/GL, then you get the genotype likelihood options, and then you can see the different models you have. So currently there are 7 different genotype likelihood models. There's version that is used within SAM tools and GATK that you saw previously, and then there's different ones like SOAP, SNP, and the Soo-Young Kim model, and one that was developed by some collaborators, some physicists. That's why we call it FUS, but these would of course not be something we would recommend. Many of these are internal tools. So okay, so if you wanted to look at the major and minor, then you can of course look at the reads, like the how many times do we observe the A, C, G, and T, and then figure out what the 2 most frequently occurring. The other thing you can do is to similar to what we did with the genotype likelihoods, then we can use those genotype likelihoods to figure out what the most frequently occurring bases are. So that would be the do major 1 option. So if we were to run this, then we would generate an output file. It can also output BCF files, and then output is just called this. So Angst put MAFS file. Here it's called the chromosome position, the major and the minor. Then it's what the frequency for the using the EM algorithm. Then it also tells us the p-value for the site being variable, and then the number of individuals with data. So if we wanted to compare this here, then we can see it's not really the same sites that we have, right? And that has to do with the SNP calling criteria. That they're calling SNPs in different ways. But if we look at this first position here, position 1, well, let's just look at the 3 last digits, 873. Then it says that it's an AG with a probability of the allele frequency is 0.27. We get roughly the same in the other one, but here we are inferring the major and the minor. This is called GA. In that case, it's called AG, and that's because it's reference alternative polarization, whereas what we have is the major and the minor polarization. So this would have been an exercise in a different context without Corona, would be to run this on a data set and then figure out what the overlap is between the SNP calling criteria and then observe the differences. Okay, so that was a brief primer on SAM tools and Angst that's going to be used a lot in the next session. But I think we should just continue here. So I'll stop sharing this screen here. Was there any question for this?
I'm not sure that everyone were able to follow.
Okay.
I guess we won't come back, you know.
Yeah.
Yeah.
Okay, sorry. If things are moving too fast with these class exercises, then within the terminal, then please let me know. My problem is that I've done this for 15 years, so I end up doing things maybe a bit too fast. Yeah, but if there's questions, then let me know. The purpose for this exercise was to be able to calculate allele frequencies. No, the real purpose was here to learn the formats and the software, and then as an example, then calculate allele frequencies and compare those. Okay, so I'll start the next slide, and this is going to be a lot more hand waving. And this is, I think this is the correct one. Can everybody see this?
Yes.
Good. Okay, so in many classic problems, oh, sorry about this. We are interested in finding some kind of maxima. We have some kind of function that defines some kind of surface, hypersurface. Most often it's a likelihood surface, and then we are interested in finding some parameters that maximizes this likelihood, or what the parameters that explains the data the most. Okay, so this is a classic situation, and if you are modeling things directly in a different context, then you can use all these numerical tools like steepest descent algorithm and Nelder-Mead algorithm, and I think it's also called the amoeba, and BFGS, the Bruyton-Fletch algorithm, and there are many optimizers. And if you're doing any kind of machine learning, then you know that there are routines for doing these things, and there's not one algorithm that's the best, because they are capturing different aspects of the data. So one algorithm might be very good for a specific function or a specific class of function, but that optimization algorithm might not work for other problems. So there's a different kind of approach to finding these kind of optima, and these are a family of iterative algorithms, where we are basing the we are generating a new estimate based on the old estimate, and then you can prove mathematically that it will improve your likelihood for each step. And this is very different from classic optimization algorithms. And I think the best way to explain this or motivate this is to look at this thing, classic algorithm in machine learning, which is called the k-means. Basically, you have in this example, it's in a plane. So we have n data points in a plane. And then you want to assign a label to each of these, such that they get associated with a specific centroid, which is the closest closest to it, right? And then it's the matter of picking these centroids. And there are many ways of picking these points. It's a NP-complete problem, but there's a very efficient approach, which is called the k-means approach. So we have this we have these points here. Then we just randomly pick 3 centroid centers. Okay, this is what we start out with. Then we are looking at for each point, then we assign those to the centroid that they're closest to. Okay, so these 3 here would be associated with this one. All these blue would be associated with this dot here, and then all these green ones would be associated with this one. Okay, and then now you have these 3 are grouped together. They share the same assignment. They're part of the same color. And then you can pick the centroid, which is in the center of all these points. So now the centroid gets shifted in this phase, gets shifted towards the center of all these. Okay. So now we have this new centroid. Okay. Then we do the reassignment again. So now we are looking at all the points, and then we see which centroid are we closest to, and then we have that these two actually suddenly shift into this one. And in those over here, they don't change assignments, and then we can do those. And then for interestingly, it works very well. And this is so here it's written more formally, right? We initialized our centroids with K, then we are assigning each point to the closest one. Then we are calculating the new centroid. This is like the indicator function. So and then the algorithm just loops between this assignment and centroid step. And for and each time you do this, then you can calculate a sort of cost function. And this cost function is just the overall squared difference that you have in your entire data set. And as your algorithm loops, continues running, then you get your cost function will be lower and lower and lower. Okay, it will continue to decrease, but it will get lower and lower and lower. Okay, so this is just a hand-waving argument that this works, and it works very well. There's of course this issue that it's very much dependent on where you start your initialization, and it might get stuck in these local minima. So of course it should be with many optimization algorithm. It depends on the starting point, so it should be redone a number of times. And then which one to choose, which clustering to choose, would of course be the one that has the overall lowest cost. Okay. Okay, so this is the, you take each point and then you do a hard call. You say it belongs to this centroid, okay? And then you are doing this kind of clustering where you shift between assigning and then recalculating, assigning and recalculating. Okay, so the EM algorithm is built on this idea. And it's so the scenario is this, that you have a very difficult function that you cannot optimize because it's intractable for multiple reasons. If you are looking at these figures here, then we can see that they are actually very nice and distributed apart, these clusters. What we could have was that we might have like different kind of Gaussian distributions, right? So that would be on top of each other. So one would be like an oval that looks like this, and then there would be a different kind of clustering that was an oval that went like this. So right, these would be completely overlapping, and then but it would actually be a mixture of two different Gaussian distributions. This would not work with the k-means. It would not be able to solve this problem. Okay, so what the expectation maximization does is that it tries to take this huge intractable optimization problem and then changes it into two-step, which can be made, that's a lot easier by assuming that you have some kind of unobserved latent variable that explains your some aspect of your data. So the classic example for this expectation maximization is a Gaussian, mixtures of Gaussian distribution and then a coin toss. So let's go through the coin toss example. You have two coins, right? Heads and tails. So in a world where nobody's cheating, then you would have that each of these have you have an equal probability of getting heads and tail, 0.5 for each of them. But this is these two coins have different kind of biases, right? There's a larger chance of actually getting a heads for one of them, and they have different biases. So the purpose is to figure out, we want to figure out what are the biases for the different coins, okay? So in the simple example, there we have the two different coins. Then we pick one of them. Then we throw the coin 10 times. Okay, we write down what we get. This is the example here. We are getting heads and tails and tails and tails, heads and heads, tails, heads and tails in this order. Okay, so this example we get 5 heads and 5 tails, and this was with the coin B, the second one. Okay, so now we pick one of these coins at random. This time we pick the first coin, and then we redo the coin throwing. Now we get almost only heads. We get a single tail. Okay, so we do this 5 times. So the data points that we have here is that we have a vector that's 50 observations long. We can call this our training data set. And then we also keep track of which coin it belongs to. And then since we know what coin it belongs to, then we can just, for each of those coins, then we can say, how many times do we observe a heads out of everything? Okay, so for coin A, we are observing 24 heads and 6 tails. So the bias, our maximum likelihood estimate of the bias for this is just the overall proportion of how many times you observe it. That is, of course, 24 divided by 30. Okay, so this will give us a bias of 0.8. Okay, so this is the parameter we seek to optimize. We seek to find. Okay, for coin B, there we are calculating how many times counting how many times we observe a head. That's 9 times, and then we observe a tail 11 times. So that would be 9 out of 20. So this will give us a different kind of bias. Okay. So one thing which is always confusing to people here is that these should not sum to 1 here, right? Because we have 2 different coins with 2 different probabilities. Right, so the probability that the A coin has a tail is 1 minus theta A. Okay, so these should not sum to 1. Okay, so this is how we would do it if we actually observed which coin we were using. So the big question here is, the problem is that what if we were just given these 50 throws and we do not really know which coin they came from? How can we attack this problem? And this is not possible to do with a classic maximum likelihood solution. You need to split this up into these 2 steps, which is called the expectation step and the maximization step. And in this very simple example, it also becomes very intuitive. First off, we are given, we start out by assuming that we have some biases, right? So this is something we completely make up, right? We assume that the bias for the first coin is 0.6 and the bias for the second coin is 0.5. Okay, and then given these probabilities, then we are calculating based on the data what is the probability of the different coins. Okay, so in this example we get that it's the probability is of that we were using the first coin is 0.45, and the probability that we're using the second coin is 0.55. Okay, so in this context, these should sum to 1. So we are getting a probability that we were using one coin or the other coin. Okay. Okay, so what we do now, this is our probability of which coin we were using given the data. What we can now do is that we can, based on this probability of the coin, then we can look at the data, and then we can recalculate a new estimate of these thetas. Okay, so given these values, then we'll end up assuming that we should have 2.2 heads and 2.2 tails. And we do this for each of these 10 observations using these distributions. And then we calculate, similar to our estimate up here, then we are calculating how many times do we expect to observe heads and tails. And then we have these new updated theta parameters. Okay, so you continue to do this, and for some reason which is not clear, this will end up converging. You're given it's given as a mathematical fact that it will converge to an optima. It might not be global, but it will converge to an optima if you run this long enough. Okay, so this was so this was the model we were using. The latent variable for this example was which coin we were using. This is what we don't observe. And then what we wanted to estimate was the bias frequency for coin A, right? So the bias frequency was not the probability that we were given coin A or B, right? Sometimes this also gets very confusing because we're always using theta as something we are we seek to optimize, and it depends on the context, if it's model parameters or if it's something that we should optimize. Or so here we seek to optimize the bias frequencies for the different coins. But what we are using in this expectation step is that we are calculating the probability that we are using one distribution or the other distribution. Okay. Okay, so I know this is super confusing. So here is a full R example that explicitly models and calculates a single of these steps. So here we are keeping track, not that it's heads and tails, but just the number of heads we get. So in this one, we have 5 heads. Here we have 9 heads. So this one is called 5, 9, 8, 4, 7. Okay. Then we are modeling this as a binomial distribution. Okay, with 10 observations. So we are calculating the density using the theta here. Okay. We call this is the likelihood, but that might not be. It's actually more like emission probabilities, I would say here, because it's conditional on the data. And then we are calculating the probability of the coins, right? So VA here we call it the weight, right? So that would be the probability of having getting the first coin for the first set of observations. And then based on these weights, then we can calculate the next step, our updated estimates. Okay, so this is the idea behind the model. There's I think 8 or 9 slides where it's being proved. We will not go through this. What I want to note here is is just the summary of it. So we have a likelihood function that looks like this, and then we want to find the thetas that maximizes the likelihood. Okay. In our case, we are not really using the likelihood, right? We are using the log likelihood, but it's still a maximization we are doing. So when we take the log of our likelihood, then this thing turns into a sum, and we can shift in the log. Okay, so what we do is that we are modeling things with a latent variable that we in the literature always call C or Z. So in this example, we just have P of xi and theta. Then we can apply the law of total probability. Then we just need to sum over all possible states. We are having some conditions that our mixing probability should sum to 1, right? If we have the probability distribution of our, and we know which latent variable we had and the mixing probability, then we don't need this mixing probability. If you know exactly for which observation we had and the theta, then we know what mixing probability it should be. And then for proving it, it's you need to use something which is called Jensen's inequality, and that basically defines that if you have a concave function, then whatever chord you're choosing, then the curve is actually above the line. So you can write down the equations, and what you do get here is that we are defining a function that we are calling Q, that we are multiplying and dividing with, and we can do this. This is allowed, right? We are just taking we are multiplying, dividing by the same, and then by applying this Jensen's inequality, then this Qi will be a lower bound of your log likelihood. So what you do get is that I'll show you here in this figure, is that you have this kind of function that we want to optimize, which is the blue one. Okay, then we define this Q function, which we know is always as good as at the specific point at our current estimate of theta, it gives exactly the same value. And we know that when we are optimizing this, then the optima for this auxiliary function Q that we have defined will improve the likelihood. So we are starting with the theta here, then we are optimizing this auxiliary function, right? That's the maximizing the step. So we are updating our new theta estimates. Then we are shifting to this next step. Then we are again defining a new Q function that looks like this. Then we are maximizing this Q function, which is over here. So we will, as we are making more and more of these steps, we'll get closer and closer to a local optima. Okay, so this is a lot of hand waving, and the reason why we are using the EM algorithm explicitly in this kind of mathematical modeling of this when we are doing NGS analysis is, of course, that we do not observe the genotype, right? These are in many cases our latent variable. We do not observe the genotypes. This is some unobserved probability measurement. So we make our latent variable these unobserved genotypes, or we make the latent variable that we are modeling in the actual the population genetic downstream analysis a function of these latent variables. So all these formulas, these are not relevant, I would say. If you are under some very specific constraints, then you can let's see, then we can apply, let's see, what's the one we're always using? Then it ends up being this kind of sum our maximization step. And that is simply the, you have a probability measure of your different kind of distribution. And then it's just the overall proportion of time that you are in these different probability distributions. Okay, so this one thing which is extremely annoying with these EM algorithms is that they're horribly slow, right? So when you're running it, it will be very, very fast at converging, right? You'll get a large increase in your likelihood at the very first steps. And then afterwards, it will converge slower and slower and slower and slower. Okay, so there was a whole family of acceleration schemes that people came up with 15 years ago. So this is not people that are working in genetics and population genetics. These are mathematicians and physicists that were looking at these EM algorithm and came up with different ways to improve it. And the one that we are using are the squared ones, the squared accelerated versions. And these are built on the idea that if you're doing one EM step, right, then you know that you are improving your likelihood. It's a better guess, right? You're getting closer to your optima. And then based on this guess, you're doing the next guess and the next guess and the next guess. And then based on the difference in parameter values between these guesses, then it's then you can use this information and do like a sort of linear combination that will allow you to jump further and further. Okay, so if you're working with a very, very high dimensional parameter space, then convergence can be horribly slow. For simple scenarios, we are seeing that it can converge in as little as 30 steps, but just with the allele frequency estimations, you do once in a while get situation where you need to optimize it for 300 iterations, and that is just a single parameter that's being optimized. So with the admixture model that we'll talk about next time, there you have hundreds of thousands of parameters that you are optimizing jointly. Right, so there you definitely need to be able to accelerate these optimizations. But it's very simple what it does. So it defines an alpha, which is just the squared difference between these successive calls, and then it's using this as a kind of weight for the next guess. Of course, there's a lot of branching operators going on. Like you need to make sure that that your step actually improves your likelihood. If it suddenly not improves your likelihood, then you might have stepped too far down your. In this example, then you are not really finding the optima, but you might have jumped all the way down here. Okay. So what we do in that case is that. Then we are not doing the accelerated step, but we are doing a single extra EM step, and then just shifting this logic with one step, and then we redo it. And this makes the optimization go from 20,000 EM steps down to maybe a few hundred. This will make a huge impact for all these analyses where you have data for each position on the human genome. You might have you have 3 billion sites on the human genome. So if you load all this information into your memory, then it's a huge block of memory that you're occupying. And if you write a function that is dependent, you have a model that is dependent on all sites on the human genome, then you need to traverse this huge block of memory. And that is what will take time. You can, of course, do threading and subdivide your data set into smaller parts and run the analysis on those, and then tries to merge the results. But what we really want is just to limit the number of time that we actually need to loop over 50 gigabytes of computer memory. So this is for, I think, for all models that we have developed in the last 7 years, we end up with these with an accelerated EM algorithm, because they're so easy once you get to the maximization step, which can be a bit tricky. It's not so that might be tricky, but you have a different problem with the classic optimization algorithm. If you're optimizing a probability measure, right, then they are not free parameters, but they need to sum to 1. Okay, so what people then do in a classic optimization algorithm is that they rewrite their function, so they suddenly have instead of optimizing n parameters, they're optimizing n-1 parameters, and then the last parameter is internally 1 minus the sum of the rest, and then suddenly you have a completely different optimization problem, because then it's then you have conditional on this, so you have a border, and then you on your then you're putting in these dependencies within your likelihood function. So this can be very, very difficult for the optimization algorithm to solve, because they are dependent with each other, and if it's using the gradient surface, then some of these parameters might be interconnected in subtle ways. Yeah, so okay, so I think this was a short primer for why we are using the EM, and I think this we should stop now. And what we will talk about next time, then we will focus a bit more on the actual application and statistical modeling with regards to the population genetic analysis. Explicitly, we will talk about the site frequency spectrum that I assume Rasmus talked about. We will define this again and talk about why it's important. I'll show you how to estimate this using state-of-the-art methods of that is currently available, and how you can do scans along the genome for finding areas of selection, and how to determine if there's some kind of population demography acting. That's one thing we'll talk about. The other thing that I'll also talk about is this admixture analysis. Based on the sequencing data, how can we properly assign ancestral proportions to the genome? This is a very interesting problem. So if you assume that I'm a mixture of person from Denmark, and from my mother might be from Sweden, and my father might be from Germany, then half my genome would be of German ancestry and Swedish ancestry, and then you can make these sorts of admixture plots where you look at the proportion of the assignment to some hypothesized ancestral population. This is very interesting. Yeah. So this is what we'll talk about the next time. Are there any questions?
I guess everyone a bit confused, because there are actually quite a lot of information
Okay.
on one slide. Well, I bet that is why they are all silent.
Okay. Okay, so for the next time, we'll skip all the math and gory details, I guess, right? I'll make sure that it doesn't get too complicated.
Oh, well, it's fine by me.
Yeah.
Well, actually, I think that's fine to have maths, even if people do not understand it right during the lecture. They can work through it later on.
Yeah, yeah. So this is the full proof of the EM algorithm, but if people have never seen it before, I don't think it's very useful to actually go through it. It might make more sense to go through it after people have read up on it. Yeah.
Well, let's then hope that by next lecture, there will be some questions.
Yes. Yeah. So if there's questions for any aspect of these things, like for the software analysis and for the terminal and how to access these things, the definitions of these basic population genetic things like allele frequencies and genotypes, then please let me know, and then we'll take it from there. But next time it will be more visual. So the results that, like the side frequency spectrum and the admixture, looks beautiful, right? It could always be an art installation when we look at these plots, right? So hopefully people will think it's super sexy. People also tend to really like this idea about assigning ancestry to individuals. So hopefully people will find that interesting.
I'm sure they will.
Mm.
All right. Thank you very much, Thorfinn.
Okay, super.
Thank you.
Yeah, and see all of you in a week.